So, having talked about the cerebral cortex, we are now going to look at the vertical systems. So, dividing the nervous system up into regions or into vertical systems or individual functional systems, there obviously is going to be a little bit of overlap. And so, some of the things we've discussed in a different light before we are now going to look at it from the point of view of vertical systems. So it includes the peripheral somatosensory system, the central somatosensory system, and the peripheral and central motor systems. And then we're going to talk about a few little things at the end of the module that influence the motor system. So with the peripheral somatosensory system, the learning objectives, we want to define some terminology. Stereognosis, we've already talked about a little bit with regard to central nervous system dysfunction. Dermatome, tract. And with the tracts, we always want to know where they go to and from and what sort of information they carry. The primary function of the somatosensory system, we want to be able to classify 6 different types of sensory neurons by size, type of receptor, level of myelination, and the type of information transmitted. And we've talked about some of these neurons before, and the functional significance of axon diameter. We've also talked about that before. We should be able to name the stimulus for mechanoreceptors, chemoreceptors, thermoreceptors, and nociceptors. Some of those you might be able to just guess from the name of them what type of information they receive. And sensory information is used for proprioception to sense body position, and there are lots of different receptors that contribute to our proprioception. And then where are the cell bodies of primary somatosensory receptor neurons located? And we'll talk about the anatomy of that. So somatosensation is sensory information from the skin and musculoskeletal systems. So information from the skin is superficial or cutaneous. So touch, pain, temperature. Information from the musculoskeletal system includes proprioception and pain. Information in the somatosensory system goes from the receptor through a series of neurons to the brain. And normally it's a 3-neuron pathway. So the first order neuron is the most distal, and it takes the information from the periphery into the dorsal horn of the spinal cord. The second order neuron travels up the spinal cord to the thalamus or brainstem, and then the third order neuron takes it from the thalamus or brainstem to the cerebral cortex for processing. The thalamus also does a significant amount of processing, and we'll talk a little bit more about that. So cutaneous sensation is information from the skin. And the speed of information processing that we're getting from our skin depends on the diameter of the axons. We've talked about this before. Larger axons transmit faster. And the degree of myelination, more myelinated axons transmit faster. Also, the other factor is the number of synapses in the pathway. It's like taking a flight with a lot of connections. It's going to take you longer to get there than a direct flight. So the more connections, the more synapses, the longer it's going to take the information to get where it's going. Sensory receptors are specialized nerve endings on peripheral sensory nerves that respond only to a specific type of stimulus. The stimulus has to be adequate to generate an action potential, and that's under normal conditions it responds to that type of stimulus. If it isn't normal conditions, you might not get the same response. Mechanoreceptors are stimulated by mechanical deformation of the receptor by touch, pressure, stretch, or vibration. And there are specific mechanoreceptors that respond to each one of those things. So one of the examples is there's one that's called a Ruffini ending, and it's like a little crumpled ending. And when you stretch the tissue, it uncrumples it, and that creates the receptor potential. Chemoreceptors respond to substances released by cells, including damaged cells after injury or infection, like we've talked about in pathophysiology. But there are also chemoreceptors in the autonomic nervous system that respond specifically to things like oxygen levels, carbon dioxide levels, glucose levels, that sort of thing, so they can adjust the internal conditions in your body to maintain homeostasis. So chemoreceptors are very important in the autonomic nervous system. Thermoreceptors transmit information regarding heat or cold. And again, they're specific to what they are transmitting. So there are specific cold receptors and specific heat receptors. The sensory receptors are located at the distal end of a peripheral nerve, and they encode whatever the physical stimulation is into receptor potential. So they transduce that physical information into neural energy that can be transmitted through the nervous system. So receptors, all of those different types of receptors can also be classified as tonic or phasic. So tonic receptors are receptors that respond as long as a stimulus is maintained. So they don't accommodate or stop responding. They don't adapt to a constant stimulus. They keep on, as long as you've got the stimulus going, they're going to respond. Phasic receptors, on the other hand, do habituate and adapt to a constant stimulus and stop responding. The brief response of cutaneous pressures after putting on a watch. I got my new Fitbit for Christmas and I put it on and it felt different from my old one. And so once my receptors habituated to that, though, now it doesn't feel different. I don't even notice it. But it's that habituation where it adapts to a constant stimulus and stops responding. The area of skin innervated by a single afferent neuron is called its receptive field. Receptive fields tend to be smaller distally and larger proximally. If you recall from tests and measures class where you did 2-point discrimination, you are going to be able to discriminate 2 points closer together on a distal portion of your body than you are on a proximal portion of your body. The distal regions also have greater density of receptors. So we can more closely distinguish between 2 points on your hand or on your fingers than you can on your trunk. So the graphic from the book, caliper A, both points are in a single receptive field, and so you're only going to perceive it as 1 point. Caliper B, there's 1 point in each receptive field, and you're going to perceive that as 2 points. So you can see that if the receptor fields are more dense and smaller, you're going to be able to distinguish more fine distinction between 1 point and 2 points. So the somatosensory first order neurons have 2 axons. They're those pseudo-unipolar neurons. The distal axon conducts messages from the receptor to the cell body, and the cell bodies live in the dorsal horn of the spinal cord. The proximal axon projects from the cell body in the dorsal horn into the spinal cord towards the brainstem. Afferents are classified according to axon diameter. So from largest diameter to smallest, it's 1A, 1B, 2, A beta, A delta, or C fibers. And yes, it is annoying that they don't use a consistent lettering, numbering, but that's just how it is. Get used to being annoyed because that's how it is. Larger diameter axons transmit information faster than smaller diameter. We know that. We've talked about that before. Larger diameter axons are more myelinated and they allow for saltatory conduction. And so that is also a factor in faster transmission. So we've talked about the A betas and the A deltas and the C fibers before with regard to modalities and pain management. And then we'll talk about the other ones and kind of their role in what happens later. So cutaneous touch is recognized as fine or coarse. Fine touch includes a variety of receptors and sensations. Coarse touch is mediated by free nerve endings throughout the skin. So something like pressure or stretch has a specific receptor that receives that information. Coarse touch is just free nerve endings. And so coarse touch tends to be things like tickle and itch and things that you're not discriminating exactly like, oh, the inside of that shirt is really soft or something like that. That's more fine touch. Cutaneous receptors respond to touch, pressure, vibration, stretch, noxious stimuli like um, chemical stimuli and temperature stimuli, and temperature to the point where it becomes noxious. The contribution of cutaneous receptors also contributes to our kinesthetic sense, like where we are in space, by stretching or increasing pressure on the skin. And also, our joint receptors tell us a little bit about where we are in space. So that's pretty interesting. We talk about the peripheral nervous system being organized in a different way. The peripheral versus the dermatome innervation. So the area of skin innervated by a single dorsal nerve root is called a dermatome. And on the left side of this picture, the blue and green colored side, that is dermatome distributions of specific nerve roots. Although cutaneous receptors are not proprioceptors, the information they gather still contributes to our position sense. So even though we don't think of them as proprioceptors, they all become part of the big picture of proprioception. Dermatomes are used to diagnose radiculopathies, where there's nerve damage and pain's going from one area to another. They're also used to determine the sensory level affected by a spinal cord injury. So for the limbs, there's the dermatomal distribution, and there's also peripheral nerve distribution. So we'll talk about areas that are innervated by a particular peripheral nerve. We talked about this a little bit in kinesiology, where the anterior part of the lower arm is innervated by the median and the ulnar nerve, and the posterior is the radial nerve. So that's a peripheral nerve distribution. And again, those are used to diagnose radiculopathy or determine the effects of a lesion, an injury, basically. Cutaneous receptors, fine touch and vibration are encoded by A beta axons. So that makes sense from what we've talked about with the A betas. A betas have a small receptive field. They are highly discriminatory. They have specialized receptor endings for particular things like pressure, stretch, light touch. Subcutaneous fine touch has larger receptive fields and is less discriminatory. Coarse touch is encoded by A delta and C fibers, and it's crudely localized touch, which is A delta, or tickle and itch, which is C fibers, which are free nerve endings. So all of those things can be nociceptive if the information being coded is potentially dangerous to tissues. But nociceptive and temperature information is encoded by A delta and C fibers, and those are all free nerve endings. So you might have a temperature receptor that is receiving some information about temperature, and once it gets to the point where it might damage tissue, then the A deltas and the A betas start getting stimulated, those free nerve endings, and the body has to do something, has to decide something anyway. The musculoskeletal system is innervated by axons that give information about what's going on with the muscles. They have Roman numerals from 1 to 4 just to make it interesting. So we have A deltas, A betas, Cs, and now we have Roman numerals from 1 to 4. And just to make it interesting, there are also A's and B's that go after those. So the large afferents are types 1 and 2. So there's 1A, 1B, and 2. And then the smaller are types 3 and 4. Just to make it interesting. Muscle spindles, there are nerve endings within the muscle that respond to muscle stretch and the rate of the length of change. So the muscle changes length, and the spindle sends messages to the spinal cord about muscle length and the rate of change. The sensory endings of the spindle respond to quick and prolonged stretches. So this will come into play. Next quarter, you're going to be talking about proprioceptive neuromuscular facilitation and techniques that you can use to increase or decrease muscle tone, and muscle spindles are all about it. So the sensory endings of the spindle, in responding to those quick and prolonged stretches, they can affect muscle contractions. So afferent messages sent back to the muscle to control muscle length change by contracting ends of the interfusal fibers inside the middle of the muscle. The center is usually non-contractile. It's just like a length measurer, if you want to think of it that way. And then the outer part of the muscle is the contractile part. And the muscle spindle sends that information to the brain and receives information back to help control muscle length. And there are all sorts of cool human tricks that you can do to fool the muscle spindle. Golgi tendon organs are nerve endings in tendons, and they respond to very slight changes in the tension of the tendon. So they respond to tension from active muscle contraction and passive stretch, which is pretty cool. Joint receptors respond to mechanical deformation of joint capsules and ligaments. So when you get a total hip or total knee replacement, they spare the joint capsule. They cut it open and they put the replacement joint in and then they sew it back together. So you still have those nerve endings, which is why you can still have good balance and proprioception after a joint replacement. The afferents associated with joint receptors are ligament receptors. Those are type 1B receptors, pretty fast, type 1. Ruffini and pisciform endings, which are Ruffini responds to stretch, so it responds at extremes of joint range, and pisciform endings respond to movement, and those are type 2 nerve fibers. Free nerve endings are type 3 and 4, and they're often stimulated by inflammation. So those ones are the ones that if you have arthritis, you're going to be stimulating those free nerve endings because you have inflammation in the joint. Proprioception is a combination of cutaneous receptors, Golgi tendon organs, muscle spindles, and joint receptors. So lots of different information from lots of different nerve endings is going into the puzzle of proprioception. People with total joint replacements, like I said, retain good joint proprioception in mid-range. So it says in the text that the joint receptors aren't critical for proprioception, but the joint capsule is maintained in the surgery, and so you still have those nerve endings. Some of them might be damaged, but most of them you still have. So large diameter axons transmit information from specialized receptors in muscles, tendons, and joints. So those 1As and 1Bs and 2s are all muscle, tendon, and joint axons. Medium-sized axons, and those are like the 2s and 3s, transmit information from joint capsules, muscle spindles, cutaneous touch, stretch, and pressure receptors. And those also include A betas and A deltas. The smallest diameter of axons convey nociceptive and temperature information from both the musculoskeletal system and the skin, and crude touch information from the skin. So lots of different information going on lots of different axons. Whoops. Peripheral nerve lesions. Neuropathy is a general term for dysfunction or pathology of one or more peripheral nerves. A complete severance of a peripheral nerve results in lack of sensation for the distribution of the nerve. You might have pain associated with it. Sensory changes are often accompanied by motor and reflex loss, and that's because of the feed-forward feedback relationship that the sensory neurons have with the motor neurons. Unfortunately, when you have neuropathy, you get some sensory loss, and the sensory loss proceeds in the order of descending axon diameter. So the first thing you lose is conscious proprioception and light touch. Then you lose cold sensation, then fast nociception, which is the A delta fibers, heat sensation, and the last thing you lose is slow nociception. So what a drag, right? You can have no sensation except pain. But it's the way it goes. Axon diameter. It's all about axon diameter. So when you have lesions in your proprioceptive pathways, it can result in ataxia. So ataxia, which is incoordination that's not due to weakness, ataxia can be sensory, vestibular, or cerebellar. So if you have lesions in your proprioceptive pathways, it's sensory or at the receptor end. If you have it somewhere in your vestibular system, it can be vestibular. If it's in the cerebellum, then it's cerebellar. There's a test that's used to distinguish between cerebellar ataxia and sensory ataxia, and it's called the Romberg test. And you will do that in your neuro rehab class next quarter. Basically, you test balance with eyes open and eyes closed, and you can tell whether or not it's coming from the cerebellum or the sensory system. Stereognosis, which we talked about before, it can be a dysfunction of the central cerebral cortex, the somatosensory cortex, or it can be a dysfunction of the pathways leading to the somatosensory cortex. So if you can't use manipulation, touch, and proprioceptive information to identify an object, it can be either central or it can be peripheral. Nerve conduction studies are used to evaluate the function of peripheral nerves. Because large diameter axons conduct the fastest, nerve conduction studies in intact nerves usually only measure the performance of large diameter axons. They put electrodes at different areas along the nerve pathway and stimulate those electrodes, and then the computer basically calculates the velocity of the nerve. And so that can be used to detect individual lesions like carpal tunnel syndrome or nerve entrapment syndromes. So that's a common study that's done for peripheral nerves.